Hello and welcome to this video tutorial. My name is Felix. I'm an Associated Principal Scientist at Certara UK, responsible for the development of the Biologics simulator. And today we're going to look into a demonstration of the therapeutic proteins capabilities within the Simcyp Simulator. But first, what are large molecules in general? As we can see in this pie chart on this slide, we have a diverse set of different large molecules. Monoclonal antibodies, shown here in blue, are the largest group and are by far the most prominent one. But we have also bispecific antibodies. We have antibody-drug conjugates, in short ADCs, and we also have other proteins, peptides and enzymes, all of which can be simulated in the large molecule facility in Simcyp. What we also have in this pie chart are oligonucleotide drugs, siRNA and antisense oligonucleotides. There is a new model in version 23 capable of simulating oligonucleotide drugs, but this will be part of a separated video. And now we're switching into the Simcyp simulator to look on screen how can we develop a compound file, what are the different models, and what are the different options. So let's get started. When we are in the simulator, we can click on File, New Compound, select a large molecule. We will have to enter a name like new mob, for instance. Save this drug file. And now we can go into the substrate position. What you will see is that similar to small molecule drugs, if you're familiar with those, we're following the ATME scheme, defining first with chem properties and then going over distribution to the drug elimination. What we will see is that our entered name is available on all the different screens, and we can also change the compound name in here. The first decision is quite important, because here we're defining whether our protein should be conjugated or not. So if you want to simulate an antibody-drug conjugate, we have to activate the with conjugate button as shown here on screen. This is something which will be dealt with in the Biologic Focus 2 workshop. Here we want to focus on a simple case of a monoclonal antibody. What is one of the most important parameters is the molecular weight. So we need to get the molecular weight right of our protein that we want to simulate, because there's a difference if you're looking at a small protein like envorvertid, or we're looking at a large monoclonal antibody. They will have a different distribution and a different clearance behavior, and this is predicted by the molecular weight, or to be more precise, by the hydrodynamic radius. And you can see the hydrodynamic radius here on the PhysChem screen. So this is a very important parameter, often not measured, and therefore we have an empirical relationship with molecular weight. However, if you have a measured value, as with every calculated parameter in the Simcyp simulator, you can open up the padlock and you can define your own value. What we also have here on screen are the fraction unbound in plasma, which might be important for smaller peptides. And what we also have is the isoelectric point and a net charge. And this is used for the renal filtration model. So it will be important for all proteins that have a lower molecular weight than 60 kilodalton. If you're moving on now to the distribution screen, what we will see is we have two different distribution models. One is the minimum PBPK model, which is as simple as it can get, because you have one lump tissue compartment, a central blood and a central lymph node compartment. Or we can decide to go with the full PBPK model, which offers a more physiological plausible structure with all the different tissues like liver, lung, heart and kidney that are represented as compartments in the model. The structure is the same as for small molecule drugs. What you can see below is a structure of a generic tissue. So that means you can see all the different processes that are accounted for in the model. So we have our inflow into the vascular space, representing the blood vessels of a tissue, and the outflow. What we are also accounting for is the lymphatic system, which is important for large molecule drugs. And what we have as distribution into the tissue, meaning distribution between the vascular space and the interstitial space, are the convective flow and the diffusive flow through the pores of the endothelial cell layer. The theory says that we have two sets of pores, small pores and large pores, and hence why we have two of those processes with the subscript S for the small pores and the subscript L for the large pores. What we are also accounting for is the endosomal uptake, represented by K-up, where we can get into the intracellular space of the endothelial cell, more precisely into the endosome. The uptake can happen from both sides, from the vascular space as well as the interstitial space, represented here by the two arrows named as K-up. In the endosome, we have the FcRn receptor being present. And that means our antibody, or our protein with an Fc domain, can bind to FcRn by a certain Kd value that is to be entered on screen. We're forming the complex, FcRn drug complex, and this complex can be recycled. So we have a fraction recycled that determines how much goes back to the vascular space versus how much is transcytosed to the interstitial space, and contributing therefore to the biodistribution, and with which rate, and this is KRC, this is the recycling rate. All the drug that is unbound can be cleared in the lysosome by the catabolic clearance pathway that is represented here, and those parameters are on the elimination screen. What we're also accounting for is endogenous IgG. So there's a competitive binding between endogenous IgG and our exogenous drug. So this is handled in the background of the model. Here we're only entering the parameters of the drug. The parameters for IgG can be found on the population FcRn IgG screen that we have here. There are all the different parameters like the sigma values, like the K-up values that we have seen on the screenshot for the endogenous IgG. So if we're moving back to the drug, then we can see the next decision we have to do is on the FcRn binding models. We have a one endosomal compartmental model that is shown here, a simplified version where we essentially have the sorting endosome at pH 6 represented in the model structure. We can also have a much more complex model structure, separating the endosomal space in an early endosome and a sorting endosome. This model also has the FcRn dynamics being included, so that means FcRn is synthesized and degraded, FcRn can be at the surface. So this model is extremely important if you're interested to simulate an FcRn antagonist, but for the normal monoclonal antibodies, it is entirely sufficient to go with the one endosomal compartmental model. The binding stoichiometry can be either 1-to-1, this is what we have seen here on this screen, or we can have 2-to-1 binding, because in reality we have two FcRn molecules that can bind one IgG or one monoclonal antibody type. So this is a more physiological relevant structure, and it becomes particularly relevant if you want to understand FcRn modifications or extended half-life modifications. If you have only one Kd value being present, it's absolutely fine to use the 1-to-1 binding model. But if you have the Kd values for the two binding steps, the 2-to-1 model is available. Endogenous IgG will follow whatever the model selection is here on screen. One of the most important parameters is the binding affinity of your antibody to the FcRn receptor, and this is shown here on screen. 728 nanomolar is the value that we're using for IgG. However, in the literature, there's a large variability of different Kd values, with a 100-fold range that has been reported. What we have developed is a little calibrator approach, shown here by the little calculator symbol. So if you have your in-house measurement, where you measure the Kd values by plasmon surface resonance, for instance, and you have a measured Kd value of, let's say, 0.845, so that means it is a higher Kd value than we have for IgG, but the IgG value measured in your assay is, I don't know, something like this, then you can see that there is certainly a difference between the value we're using in Simcyp and the value that has been measured in your in-house assay. We're using this very simple calibrator approach to calibrate the measured Kd value of your test compound towards our value for endogenous IgG. So if you click on enter, then you can see that the calibrated value will be taken into account. Why is this important? This is important because some of the cellular dynamics are not known. The KRC value, for instance, is not measurable. So what we have done is to use IgG plasma concentration to fit the KRC value in the model. But for the fitting, we used our KD value for endogenous IgG of 728 nanomolar. So if the KD value in your assay is different, you would need to adjust the KSC rate in order to simulate your antibody appropriately. Instead of doing that, we're recommending to calibrate the KD value towards the model. What we also have is this KSC rate here on screen, which is tissue independent, and as said, it has been fitted towards the IgG data in the lack of data. Below we have then the different tissue-specific parameters. These are the lymphatic reflection coefficient that determines how much of the drug moves from the interstitial space into the lymphatic system. Very often this value is assumed to be 0.2. In the simulator we're having a value of 0 because the endothel surrounding the lymphatic vessels is very leaky with high pores, so there seems to be no evidence that an antibody cannot distribute into the lymphatic system from the interstitial space, and hence why we don't have any restriction on that. What we're also having are the two pore parameters. These are the PS values for the diffusive process, bidirectional between the vascular and the interstitial space. And we're also having the vascular reflection coefficients that are used for the convective term in the model. Those parameters, as you can see by the calculator symbol, are calculated to pore parameters. The most important factor is the hydrodynamic radius that we had on the PhysChem screen, and is the size of the pores that you can find in the population tissue pore size tab. So here you have the size of the pores in nanometer and the amount of small pores versus large pores, which is also important in the model. So what we do is looking at the hydrodynamic radius. So if we have a typical monoclonal antibody, it would be roughly 5 nanometer. And then we are looking at it with the pore sizes that we have. So for instance, in adipose tissue, we have a small pore size of 8 nanometers, so the drug would be able to distribute through those pores. But in brain, we just have 1 nanometer, so there would be a restriction. There would be no flux from the vascular to the interstitial space in the brain compartment. We do this for the small pores, and we do this for the large pores. There are other factors that are important, like the osmotic pressure gradients that are used then in the terms to calculate the diffusive process. What is also important are the lymph flows, and the lymph flows can be found in the tissue flow screen of the population. So we have here the percentage of total lymph flow rate for the different compartments, and next to it in gray there are the lymph flow rates of a population representative for your orientation to see how much of lymph flow we actually have. On the lymph tab you find the total body lymph flow rate that is used then to calculate the regional lymph flow rates and the lymph volume. If we're moving back to the distribution screen, then what we will see is that we have the parameters of the uptake rate, the endosomal uptake rate, as well as the fraction recycled that determines how much goes vascular versus how much goes to the interstitial space. Again, those values are extremely difficult to measure. For the uptake rate, we can use pinocytosis markers like horseradish peroxidase, but the question is, how representative are they of a monoclonal antibody? And a more important question is, how does it differ between different locations in the body? Is an endothelial cell in the liver similar to an endothelial cell in the kidney? And the other question is, how different is it for different antibody types? And one of the factors that can have an impact is charge, because our plasma membrane is negatively charged. So a positively charged protein would have electrostatic interactions with the plasma membrane, and therefore a positively charged protein can have a higher likelihood of getting in the endosomal, into the endosomal space. What is very important to mention is that the uptake rate is the bottleneck for the FcRn recycling pathway. Everything that gets into the endosome can bind to FcRn or can be cleared via the catabolic clearance pathway. So knowing K-up becomes really important, and knowing the effect of charge on K-up becomes important. Unfortunately, today there are no in vitro assays, or there are heparin columns, but they are not correlated to the cellular uptake of known proteins, so we don't have any relationship at the minute, but something to keep in mind when developing the compound file. With that, we have looked at the different distribution parameters, and now we move on to the drug elimination screen. The drug elimination screen contains the total body catabolic clearance in the lysosome. So this is again a value that has been fitted towards the IgG plasma concentration, but is backed up by experimental data that suggest a half-life of a protein to be 8 minutes in a lysosome, and a lysosomal volume that is 1% of total cell volume, and that leads to roughly the catabolic clearance we see on screen. What we also have is the fraction of catabolic clearance that is shown here below. That distributes the total body catabolic clearance to the different compartments of the model. And this data have been published and have been measured in mice together with in silico modeling. What we also have is an additional systemic clearance that drains the drug from plasma in a linear way, and it describes all the pathways that we are not accounting for in the Simcyp simulator. So that might be uptake by non-FcRn expressing cells with subsequent degradation, or it might be any interaction with immune cells like macrophages that are not modeled in the Simcyp model. What we're also having are different tabs for renal filtration, which is deactive by default. It would not play a role for our monoclonal antibody, but it definitely plays a role for smaller proteins and peptides. And then there's also the possibility to enter tissue metabolism if that is measured, for instance, in human liver microsomes for peptides, as it has been published in the literature. The next point that we like to look on is the TMDD model. This is the binding to our target of our antibody. By default, TMDD is deactivated, so we can activate TMDD. And then there are different models that we can use. One-to-one binding of our drug to the target. We can again have a two-to binding stoichiometry if those binding affinities are known. We can model bispecific antibodies. Again, this is a focus of the Biologic Focus Workshop 2. And we can have target shedding. In this case, we're going now with a one-to-one model. And then we can select on different options that we're going to look in a minute, because it will change the way we're looking at parameters. First, we're defining a target by pressing this plus sign. So we're getting a target in here. You can see there's a huge list of different targets. Some of them are predefined. They come with our implemented compound files. Some of them are flexible to define. So we're taking one of the more flexible approaches. We can decide where the target is expressed. For instance, we say it's expressed in the liver, so we click this little icon and activate the liver button. If we say our target is expressed at two different locations with a different abundance, we can again add the plus sign, select on the target number 10 and say this is for the kidney expression. So you can see the target can just be at one location. There can be only one target, but we can define different targets at different locations. Now we have our binding parameters. So if you're selecting the full TMDD model, we have to enter an association and a dissociation rate constant. And this model selection is the same independent of how many targets we have chosen. If we have only a KD value, we can use the quasi-equilibrium approximation that assumes an instant equilibrium at beginning. There are also two other model choices, quasi-steady state, which also assumes an equilibrium between the binding and the internalization, and we have a Michaelis-Menten model that's more familiar for population PK modeling. There's also an option for a constant Rmax, which means the internalization rate is grayed out, similar to the degradation rate of the target, or it can be dynamic. And then we can start to enter our values here. But how do we define the target abundance in general? And therefore we have a little go-to button below here, which we can click on. It moves us to the population target screen, and here we can start to define our target. So we can call this new target liver, and we can call this new target kidney. And now we can start to define the abundances and say, okay, maybe it's 0.01. And that means we can have a different abundance. So if we know it's expressed at different locations in the body, but it has a different abundance, then we can use the system to define multiple targets, to define it to specific locations, and to give it a different abundance. And then the degradation rate might be the same. So we have to enter a degradation rate, and the synthesis rate will be automatically calculated based on the simple turnover model. What we also need in this case is the molecular weight to do the unit conversions, because the simulation runs in micromolar. If you would like to have the outputs in a mass unit, we need to have the molecular weight of the target in order to convert the target concentration and the drug-target complex. With this, we have set up also the TMDD model. One word to the TMDD, it can be linked into the PD models. So we can activate a PD model. We can select, for instance, the liver concentration. And that means we can also select the drug-target complex or the free target concentration to drive our PD effect. So there can be a link between the PK and PD. One word on the outputs that we're having. They are very similar and familiar for those of you who are familiar with the small molecule simulator. What we have in addition are for the drug-target binding models, we have something like target status that defines the abundances of our target and defines the synthesis rate for each individual. And then we have the TMDD profiles that look into the concentration of the free receptor, the receptor occupancy, and the concentration of the drug-target complex. One last word on the trial design or the different routes of administration that are available. We have IV modeling, we have first order absorption, we have subcutaneous and intramuscular absorption, we have an intrathecal brain model, and we can also have custom dosing to combine different subcutaneous administration sites. This concludes the little round tour of the Simcyp Biologics Simulator. I hope I could show you today some of the basic principles of the model, some of the important parameters that you require to develop a protein in the simulator, that are the hydrodynamic radius. We need to have an idea about the clearance, we need to have an idea about the binding affinity to FcRn, and if TMDD plays a role, we need to have an idea about the target, its location, its expression, its degradation, and the binding affinities. With that, I'd like to thank you for watching this video. If you are interested, please visit one of our biologic focused workshop. The biologic focused workshop 1 will deal with the principles of the large molecule simulator. We will go through those, what I've mentioned, in more detail. And in the biologic focus 2, we go to more complex biologics. That means antibody drug conjugates and bispecifics. So thank you very much for watching this video,